It is late, and you have arrived here. Whether your day was kind or difficult, this hour is yours to rest. Let the small worries that climb into the dark slip back beneath the surface. Tonight we will drift very slowly inward, past your skin, past your blood, past the soft walls of a single cell, into the quiet nucleus, where the long patient library of you is gently spooled and waiting. Welcome to the Circadian Institute. Each night we go somewhere far from the noise of the day, somewhere ancient, somewhere unhurried, somewhere ready to receive a tired mind. Tonight that place is the slow molecular library that lives inside every cell of you, the double helix, the 4 quiet letters, the patient copying, the gentle reading, the long ancestry of every living thing. A subscription is the quiet way to stay with us for tomorrow's journey, and the one after. There is nothing to learn tonight, nothing to remember. The story of DNA is the quietest story there is, written in only 4 small letters, copied very slowly, generation after generation, for almost 4,000 million years. You only have to rest and let the slow reading move past you. We will drift softly from the first idea of inheritance through the quiet shape of the double helix, the 4 patient letters, the slow copying, the gentle reading, the long ancestry inside every living thing, and the long quiet library that is, very softly, all of you. No alarm, no memorizing, only the quiet of molecules. Settle in now. Close your eyes if you would like. Let the voice be a soft current, carrying you inward into a single patient cell. You will not need to be awake at the end. Sleeping is the right answer. Long before anyone knew about cells, people knew quietly that children look like their parents. The eyes of a grandmother showing up softly in a grandchild. The quiet way a calm dog passes a calm temperament on. People knew, without knowing how, that something was being handed down. For most of human history, this passing down was a kind of gentle mystery. People imagined small unseen seeds, or thought the blood itself carried the family resemblance. They were not wrong about the softness, just about the shape. In the 1860s, a quiet monk named Gregor Mendel grew small pea plants in the garden of his monastery and counted them patiently. How many were tall, how many short, how many had round seeds, how many wrinkled. He kept very careful notes. From those quiet counts, Mendel saw a pattern. The features of the parents did not just blend together. They were passed on as small, separate, quiet units, each one either there or not there, kept whole across generations. Nobody really noticed Mendel's quiet work for almost 40 years. The small units he had imagined would later be called genes, but for a long, calm time, nobody knew what a gene was actually made of. Slowly, very slowly, biologists began to look inside cells. Under quiet microscopes, they could see small dark threads inside the cell's nucleus. These threads, when a cell was about to divide, would line up gently and split, half going one way, half the other. These dark threads were named chromosomes, colored bodies from the way they took on dye in the soft staining experiments of the time. People sensed gently that the inheritance lived inside them, but chromosomes were made of 2 things, protein and a quieter molecule called nucleic acid. For decades, most people quietly bet on the protein. Proteins seemed complicated enough to carry the long story of life. The nucleic acid, a simpler, quieter molecule, seemed too plain to hold so much. Then, in 1944, Three quiet scientists in New York, Avery, McLeod, and McCarty, did a patient experiment with bacteria. They showed that the thing being passed from one harmless bacterium to another and changing it was not the protein at all. It was the quiet nucleic acid. The plain molecule was the message. That nucleic acid had a quiet, long name, deoxyribonucleic acid, for short DNA. From this moment, the search began for what shape it had, because the shape of a molecule very softly tells you how it works. In London in the early 1950s, a careful young scientist named Rosalind Franklin was taking X-ray pictures of DNA, patient, exposure after long exposure, capturing the soft way the molecule bent the light. Her photographs were the most careful in the world. Her clearest picture, called Photo 51, showed a soft, gentle X shape with quiet, dark spots arranged in a way that could only mean one thing. The molecule was a spiral, a patient, twisting shape, like a long, quiet ladder gently turning around itself. Two other scientists in Cambridge, James Watson and Francis Crick, saw her picture, and very quickly, in the spring of 1953, they built a small model of the molecule out of cardboard and metal. The shape they made matched the patient X-ray picture exactly. The shape was, very softly, a double helix. Two long strands wound around each other like a slow, patient staircase. Inside, between the two strands, were small flat units called bases, stacking like the steps of the staircase, one above the other. It was a calm, beautiful shape, and the moment people saw it, they understood gently how the molecule could copy itself. If you pulled the two strands softly apart, each one had all the information needed to rebuild the other. The shape itself explained the inheritance. That is the quiet idea we will follow tonight. Inside every single cell of every living thing, inside every cell of you, there is this patient, twisted molecule holding the long, slow, soft instructions for being alive. And it has been there, almost unchanged in its shape, for almost 4,000 million years. Picture, very softly, a long, slow spiral staircase. Two outer rails winding gently around each other. Between the rails, small flat steps stacking patiently, one above the next. This is the quiet shape of DNA. The two outer rails are made of small repeating pieces. A sugar molecule called deoxyribose, joined to a small piece of phosphate, joined to another sugar, joined to another phosphate in a long, calm chain. Sugar, phosphate, patient, regular, quiet. Two of these long sugar and phosphate chains run gently alongside each other. But, and this is part of the quiet elegance of it, they run in opposite directions. One chain points one way, the other chain points the other way. They are soft mirror images. Between the two chains, like the soft steps of the staircase, are small, flat, ring-shaped molecules called bases. Each base is joined to a sugar on one chain and reaches gently across to a base on the other chain, where the two soft bases touch. There are only 4 kinds of base in all of DNA, only 4. The whole long, quiet library of every living thing on Earth, every tree, every whale, every fungus, every soft sleeping creature, every part of you, is spelled out in just these 4 small letters. Their names are adenine, thymine, cytosine, and guanine. Most of the time, very gently, they are just called by their first letters, A, T, C, and G. Those four soft letters are the whole alphabet of life. The way they fit together is what makes the shape work. Adenine, A, always pairs very quietly with thymine. T, cytosine, C, always pairs very softly with guanine, G. Every soft step of the staircase is one of these two patient pairings, A, T, G, C, nothing else. The two bases in a pair hold each other gently with small soft links called hydrogen bonds. These bonds are weak enough that the strands can be carefully pulled apart when needed, but strong enough that the staircase quietly holds together while it is just sitting in the cell. Then the whole long ladder, with its sugar rails and its base steps, gently twists. About once every 10 steps, it makes a full slow turn around itself. This is why the shape is called a double helix, 2 helices, 2 slow spirals, gently winding. If you could see a single tiny stretch of DNA up close, it would look like a soft turning ribbon, lit from within. The outer rails would glow softly blue. The base pairs inside would glow softly amber. The whole molecule would seem to be very, very patient. This shape is the same in every living thing on Earth. A grain of wheat, an oak tree, a slow whale in the deep sea, a quiet bacterium in soil, a sleeping child. Every single one of us is holding, in our cells, the same gentle, twisted shape. The shape itself is part of how the molecule does its quiet work, because the two strands hold information in a soft mirror, A on one side, T on the other, C on one side, G on the other. You only need one strand to know what the whole molecule says. The other side is implied. That soft mirror is the secret of inheritance. When a cell wants to copy its DNA, it gently pulls the two strands apart. Then, on each open strand, the small loose bases floating in the cell can quietly find their soft match and join in. A finds T, G finds C. And from one quiet double helix, 2 patient double helices are made, each one identical to the first. This is the slow magic of DNA. The shape is the message, and the shape is the way the message is copied. There is a soft elegance to this. The whole long alphabet of life is only 4 small letters. The whole way the letters are held in place is one quiet shape, and the whole way the shape passes itself on is just 2 soft strands gently coming apart. Tonight, inside every single cell of you, there are about 2 meters of this soft, twisted ladder, very carefully folded into a space less than 1/10 of a millimeter across. 2 meters, inside something you cannot quite see. The patient packing of it is its own quiet story, and we will come to it. There are only 4 letters in all of DNA, 4 small molecules that, in their soft, patient arrangement, spell out everything from the color of a leaf to the way a heart muscle gently beats. 4, that is the whole alphabet. Their full names are adenine, thymine, cytosine, and guanine. We will mostly call them by their first letters tonight, A, T, C, G, the 4 quiet letters of life. Each letter is a small, flat, ring-shaped molecule made of just a few carbon and nitrogen atoms. They are tiny, far too small to see, even with the best ordinary microscope. But they are very patient. They hold their shape for a long, calm time. 2 of the letters A and G are slightly larger. They have a double ring in their soft shape. We call these 2 the purines. The other 2, T and C, are slightly smaller. They have a single ring. We call these the pyrimidines. The way they pair is gentle and exact. A double ring A always pairs softly with a single ring T. A double ring G always pairs softly with a single ring C. So in every soft step of the ladder, one big letter and one small letter meet in the middle, and the whole ladder stays the same width all the way up. This pairing is held together by tiny soft links called hydrogen bonds. A and T are held by 2 of these soft bonds. G and C are held by 3. So G and C pair a little more strongly than A and T. It is the smallest possible difference, and yet it matters. Because there are only 4 letters, you might think the messages must be very simple, but the long, quiet sentences of DNA are extremely long. Inside every cell of you, there are about 3,000 million letters, gently arranged in a particular order. 3,000 million letters is enough room for everything your body needs to know: how to build a heart, how to make a tear, how to digest food, how to grow a fingernail, how to sleep at night. It is the whole quiet recipe for you, written in just 4 letters. If you printed out all the letters of your DNA in a very small font, one letter after another, you would have about 1 million pages of ATCGA, library of about 3,000 thick books, all of it inside one quiet cell, all of it copied every time a cell divides. Most of those letters are the same in everyone. About 99.9% of your DNA is the same as the person next to you. Only about 1 letter in 1,000 is different across that small, soft difference. All the gentle variety of human faces is written. And about 98% of your DNA is the same as a chimpanzee. About 85% is the same as a mouse. About 50% is the same as a banana. The same 4 letters in slightly different patient arrangements spell out all of life. The order in which the letters appear is what carries the message, just like the same 26 letters can spell a love poem or a recipe. The same 4 letters can spell out an oak tree or a whale or you. The order is everything. Inside each cell, the letters of your DNA sit, very patiently, in a particular order that you inherited from your parents, half from one parent, half from the other. The order does not change much over your life. It is, in a quiet way, the longest thing about you. Some stretches of the letters spell out instructions for making specific small molecules called proteins, and we will come to those later. Other stretches do not spell out a protein at all, but help control when other stretches are read, and some stretches just sit there quietly, very old from long ago. All of this is happening very quietly right now in trillions of cells inside you. The 4 letters are sitting in their patient order. They are being read in some cells, copied in others, gently maintained in nearly all. You do not have to think about any of it. There is something very calming, perhaps, in knowing that the whole long story of you is written in only 4 small letters. 4. And tonight, while you rest, those 4 letters are doing exactly what they have always done. They are quietly, patiently being themselves. Most of the DNA in your body lives in one quiet place, a small round room inside every cell called the nucleus. The nucleus is soft and warm and gently surrounded by its own thin membrane, like a quiet inner room within an already quiet house. A cell is already extremely small, about 1/10 of the width of a human hair, but inside that cell the nucleus is smaller still, usually about 1/10 of the cell's own width, And inside that small, soft room, gently coiled, is 2 meters of DNA. How does 2 meters of soft thread fit into something 1/10 of a millimeter across? Very, very carefully. The DNA is not loose. It is gently wound around small protein spools, then the spools are wound around each other, then those bundles are wound again. It is all soft, careful packing. The nucleus itself is a very still place. It is surrounded by a soft double membrane with small gentle openings called nuclear pores. Through these pores, certain small molecules can slip in and out, but the DNA itself never leaves. It stays quietly inside. Inside the nucleus, the DNA is not in one long single strand. It is in several quieter pieces called chromosomes. We will come to chromosomes very soon. For now, it is enough to say that the DNA is gently divided into several long, calm packages. Around the chromosomes, inside the nucleus, there is a soft, warm fluid called nucleoplasm. It is mostly water, with many small drifting molecules, enzymes, building blocks, helpers, all moving very slowly through their patient, quiet task. There is also, inside the nucleus, a small darker spot called the nucleolus. It looks like a quiet pebble in the middle of the room. Its calm job is to build the small protein-making factories called ribosomes. We will meet ribosomes much later tonight. Almost every single cell in your body has a nucleus, and almost every single nucleus contains the full, complete library of your DNA. Every cell has the whole instruction set. Every cell, the skin on the back of your hand, the cells lining your stomach, the cells in your eye. There are a few quiet exceptions. Your red blood cells, for instance, gently push their nucleus out as they grow up because they need every bit of space for carrying oxygen, so they have no DNA at all, just the patient work of breathing. In most other cells, though, the nucleus is the quiet center of things. When a cell needs to make a protein, the message comes from here. When a cell needs to divide, the long, careful work begins here. The nucleus is the gentle library, and the rest of the cell is the slow, patient workshop. The membrane around the nucleus is soft enough that when a cell is about to divide, it can quietly dissolve, letting the chromosomes line up gently in the middle of the cell. Then, after the division is done, the membrane forms again around each new daughter nucleus. So the soft library is a temporary structure. It comes apart gently when needed and reforms gently afterward. The DNA itself, however, never leaves. The chromosomes simply hold together and travel where they need to go. All of this happens in extreme quiet. There is no sound in a cell. There is no light in any way you or I would recognize. There is only the soft, slow drift of molecules in warm liquid, very patiently doing what they have always done. The nucleus is also where many of the cell's quietest decisions are made. Which stretches of DNA should be read right now? Which should be quiet? Which should be copied? Which should stay still? All of it is decided here, gently, every moment. Your body is made of about 30 trillion cells. Most of them have a nucleus, so most of them have the full library of your DNA inside. 30 trillion small libraries, all of them very softly identical, all of them carrying you. Tonight, in every single one of those quiet nuclei, the soft spooled DNA is sitting calmly. Some stretches are being read, most are resting. The little nuclear pores are letting messages in and out at their own gentle pace. The library is open. The library is always open. Inside the nucleus, the long, soft thread of your DNA is not one continuous strand. It is gently divided into 46 patient pieces, each one called a chromosome. 46 soft packages, each holding a long, quiet stretch of letters. 46 is the number for human beings. Other creatures have different numbers. A dog has 78, a goldfish has 100, a pea plant has 14. The number itself does not really matter. What matters is the patient pattern. Of your 46 chromosomes, exactly half came from your mother and exactly half from your father, 23 from each. They sit very gently in matched pairs inside every cell of you, 23 soft pairs all your life in almost every nucleus. Each pair carries the same kind of information, gently doubled, one copy from your mother, one from your father of every instruction you carry. If the copy from one parent has a small soft difference, the copy from the other can often quietly make up for it. 22 of these pairs look the same in everyone, and we call them the autosomes. They carry most of the long, quiet instructions for being human, for growing, for breathing, for the soft chemistry of your blood, for the slow, steady beat of your heart. The 23rd pair is the soft pair that is different. We call them the sex chromosomes. They come in two patient shapes, a larger one called X and a smaller one called Y. The X is softly larger because it carries more genes. The Y is gentler and quieter. Most people who develop as biologically female have two X chromosomes. Most people who develop as biologically male have one X and one Y. There are also, more quietly, several other patient combinations, and all of them are simply human. Most of the time, you cannot see chromosomes at all. They are spread out very softly through the nucleus, gently uncoiled, like long, quiet ribbons floating in warm fluid. In this state, they are doing their daily quiet work, being read, being maintained. It is only when a cell is about to divide that the chromosomes gently coil up tight, so they can be safely separated into the two new daughter cells. In this coiled state, each chromosome becomes the patient X shape that you have seen in pictures. The X shape is actually two soft copies of the same chromosome, joined gently at a single point called the centromere. Each arm of the X is one full copy. When the cell finally divides, the two copies separate calmly, one going to each new cell. The longest chromosome in you is called chromosome 1, and it is about 250 million letters long. The shortest, chromosome 21, is about 48 million letters long. Each one is patient and slow in its own way. Each chromosome carries hundreds or thousands of separate instructions called genes. A gene is a small, soft stretch of DNA that spells out, in its patient order of letters, how to build one particular small molecule, usually a protein. In all 46 of your chromosomes, there are about 20,000 of these gene instructions, 20,000 small patient recipes for building 20,000 different proteins, each one of which does some quiet, specific job inside your cells. Most of the DNA, however, is not gene. Only about 2% of your 3,000 million letters actually spells a protein. The other 98% does other quieter jobs, controlling, organizing, or simply resting there from long ago. All 46 chromosomes have been in nearly every cell of you since you were a single fertilized cell at the start of your life. As your cells have divided, every new cell has received the full, careful, soft set. Every cell carries the whole library. Tonight, while you rest, your chromosomes are sitting quietly in their soft, uncoiled state, gently doing their long, patient work. Some genes are being read, most are not. The whole library is, as always, just being itself and being you. 2 meters of DNA inside a cell less than 1/10 of a millimeter across. That is the patient packing problem the cell has solved, gently in every nucleus of every living thing on Earth. If the DNA were just loosely floating in the nucleus, it would tangle hopelessly, so the cell uses a soft, careful system of spools and folds, gently stacking the long thread on itself many times, like the most patient origami in the world. The first level of packing uses small round protein clusters called histones. There are 8 histone proteins in each cluster, gently grouped into a soft round shape. They look very softly, like a small ball. A short stretch of DNA, about 150 letters long, winds gently around each one of these histone balls, almost 2 full slow turns like soft thread around a small spool. The combination is called a nucleosome. If you could see a stretch of DNA up close with all its patient nucleosomes, it would look like a long, quiet string of soft, round beads. Bead, then a short stretch of bare thread, then another bead. This is called the beads on a string arrangement. Then those beads on a string coil softly together into a thicker fiber about 30 nanometers wide. The nucleosomes fold gently against each other, forming a quiet zigzag or a soft solenoid shape, depending on the gentle context. Then this thicker fiber loops very softly into long, quiet loops that are anchored to a soft inner protein scaffold, loop after slow loop, gathering together into the larger structure of a single chromosome. In the resting nucleus, most of the chromosomes stay in this soft, loose, looped state. The DNA is still gently accessible. Reading molecules can come along and unwind small sections at a time to do their patient work. Only when the cell is preparing to divide do the chromosomes coil even more tightly. The loose loops fold against themselves again and again, gently compressing the long thread into the familiar dense X shape of a fully condensed chromosome. In its most compact form, the DNA is folded so tightly that the 2 meters of soft thread fit into a structure only a few micrometers long. That is a packing ratio of about 10,000 to 1, 10,000 times shorter than the unfolded thread. The histones themselves are not just passive spools. They carry small, soft chemical marks on their gently waving tails, and these marks help control which stretches of DNA are open for reading and which stretches are kept gently silent. A mark that softly opens the chromatin makes the DNA more readable in that region. A mark that softly closes the chromatin keeps that region quiet. The cell uses these gentle marks to decide, moment to moment, what to read and what to rest. This patient packing is happening in every nucleus right now. The chromatin is being gently opened in some places, gently closed in others. The histones are being marked, unmarked, marked again. The slow, careful library is being curated continuously, quietly. It is one of the most elegant systems in biology. a 2-meter thread packed into a space 1/10 of a millimeter across, and yet still gently readable at any point, at any moment, without ever tangling. The cell has been solving this problem for almost 4,000 million years. And yet none of it requires your attention. You are not running this. You do not have to fold anything, mark anything, open anything. Inside your cells, the patient packing is doing what it has always done while you rest gently in the dark. Tonight, in trillions of soft round nuclei inside you, the long quiet thread of your DNA is folded with infinite patience, beads on string, then thicker fiber, then long loops, all gently arranged. The library is quiet. The library is being kept. If you could very gently take all the DNA out of a single one of your cells and lay it end to end, it would be about 2 meters long, 2 meters inside something 100 times too small to see. Now consider how many cells you have: about 30 trillion, 30,000 million cells, each one with about 2 meters of DNA quietly folded inside. If you took all the DNA from all your cells and very softly laid it end to end, the line would be about 60,000 million kilometers long. That is about 400 times the distance from the Earth to the Sun. Or, to say it another way, the DNA in your body all laid out would stretch from the Sun to Pluto and back twice over. The quiet length of you, if it were unrolled, is solar system scaled. And yet the entire thing is very calmly folded inside the soft space you take up in this bed tonight. None of it is rattling around. None of it is bunched. It is gently arranged, every cell, every nucleus, every chromosome. How is it kept tidy? Through the patient packing we have just talked about. Histones spool it gently, loops anchor it to soft scaffolds. The chromosomes are kept separate, each in its own quiet region of the nucleus. If you could see a single one of your chromosomes fully unrolled, it would be about 4 centimeters long, 4 centimeters of soft molecular thread, folded into a structure you would need a microscope to see it all. Most of that length is the quiet sugar and phosphate backbone, with its patient base pairs stacked along it, 3,000 million base pairs, in each cell, in the same calm patient order, doing the same calm patient job. Reading 3,000 million letters of DNA out loud, 1 letter every second, would take about 95 years. 95 years of continuous gentle reading. The whole quiet human instruction set is longer than a human lifetime, said aloud. Almost all of those letters were inherited from your parents, half from each. Almost none of them have changed since you were a single cell at the start of your life. The library you carry now is the library you started with. And the library is duplicated. Almost every cell of you has the same complete 2 meters of DNA. The cells of your eye, the cells of your liver, the cells inside the bone of your wrist, every one of them holds the same long, calm thread. If you took a single hair from your head and gently looked inside one of its cells, you would find the same instructions for growing a tongue, or building a kidney, or beating a heart. The same library in every cell, quietly complete. Most cells, of course, only ever use a small fraction of that library. A cell in your liver does not need to read the instructions for an eyelash, so most of the DNA in any one cell is gently kept quiet, present but not being read. This quiet, selective reading is how a single fertilized cell becomes all the soft variety of you. The same library, read in different patient sections in different places at different times, gives a brain cell its shape, a skin cell its softness, a heart cell its slow beat. 60,000 million kilometers of DNA in 30 trillion cells, gently doing exactly the right thing in each place. Tonight, all of it is in steady, quiet operation while you rest in the dark. There is something quiet and reassuring in this. The library of you is vast and gently folded and patiently held, and it does not need you to do anything tonight. It only needs you to rest while the long, calm story continues to be read. Every time a cell in your body divides, it has to make a fresh, soft copy of all of its DNA. 2 meters of patient thread, perfectly copied in just a few hours with almost no mistakes. This is one of the quietest miracles inside you. The copying begins when the cell decides very gently that it is time to divide. Small, soft signals tell the nucleus to begin. Quiet molecules called helicases gather along the DNA and start their patient slow work. Helicase is a small ring-shaped protein that slides along the double helix and gently breaks the soft hydrogen bonds between the base pairs. As it moves, it pulls the 2 strands of DNA softly apart, like a patient zipper opening. Behind the helicase, the 2 strands of the DNA are now soft and exposed, each one ready to be copied. Each strand will serve as a gentle template for building a new partner. The molecule that does the copying is called DNA polymerase. It is a small, soft enzyme that crawls along each single strand, reading the letters one by one and adding their gentle matching partners on the new strand. As polymerase moves along, it picks up loose nucleotides from the soft cellular fluid around it. If the template letter is A, the polymerase quietly adds T. If the template is G, the polymerase adds C. The pairing rule is followed every time. Polymerase is also a careful proofreader. After it adds each new letter, it gently checks to see if the pairing is correct. If it has accidentally added the wrong letter, it pauses, removes the mistake, and tries again. Quiet, patient, careful. Because the 2 strands of DNA run in opposite directions, and polymerase can only work in one direction, one of the 2 new strands is built smoothly and continuously, while the other is built in small, soft segments that are joined together afterward. The smoothly built strand is called the leading strand. The segmented one is called the lagging strand. The little segments on the lagging strand are called Okazaki fragments, named after the patient Japanese scientist who first noticed them in the late 1960s. Another small enzyme called ligase comes along quietly behind the polymerase and softly glues the Okazaki fragments together, making the lagging strand into one smooth, continuous new copy. All of this happens at many places along the DNA at the same time. The whole human genome, 2 meters of soft thread, is copied at thousands of these little replication forks simultaneously, so the whole job can finish in just a few calm hours. When the copying is finished, each of the original 2 strands of the parent DNA has a new partner strand wound softly beside it. 2 full double helices now exist, where before there was one, the library has been gently doubled. Each of the two new double helices has one strand from the original parent DNA and one strand that was newly built. We call this semi-conservative replication, half conserved, half new. Each new copy carries a small piece of the original with it. The error rate of all this gentle copying is astonishingly low. Polymerase, with its quiet proofreading, makes about one mistake in every 100 million letters. Then a separate repair system catches most of those, bringing the final error rate down to about 1 in 10 billion. One mistake in 10 billion, about one tiny soft change every time an entire cell's worth of DNA is copied. Almost perfect, but not quite. And that very small almost is very gently where evolution gets its slow material. Tonight, in many cells of you, especially in your skin, your gut lining, your bone marrow, this quiet copying is happening right now. Helicases opening, polymerases reading, ligases sealing. The long library is being faithfully duplicated while you rest in the dark. DNA is the long, quiet library, but it never leaves the nucleus, so when a cell needs to use one of the soft instructions in its DNA, it cannot just send the DNA itself to do the job. Instead, it makes a small temporary copy. The small temporary copy is called RNA, ribonucleic acid. RNA is very similar to DNA, but with a few quiet differences. It has a single strand instead of a double helix. Its sugar is called ribose instead of deoxyribose. And in place of the letter T, RNA uses a soft letter called U. The process of making an RNA copy of a stretch of DNA is called transcription, from the Latin word transcribere, to write across the cell is very gently writing a soft copy of one passage of its library. Transcription is done by a small enzyme called RNA polymerase. It is similar in shape to DNA polymerase but quieter. It binds gently to a particular spot on the DNA called a promoter, which marks the start of a gene. Then RNA polymerase opens a small soft bubble in the DNA, just where it sits, only about a dozen letters wide. The two DNA strands separate gently right there, exposing the template strand. The polymerase begins to read. As RNA polymerase reads each letter of the template DNA, it picks up the matching RNA nucleotide from the fluid around it and links it gently to the growing new RNA strand. If the template letter is A, it adds U. If the template is C, it adds G. If the template is C, it adds A. The RNA strand grows out from the enzyme very slowly. The DNA closes gently behind it. The polymerase moves on. It is a slow patient writing across, one soft letter at a time, deeper and deeper into the gene. When the polymerase reaches a special soft sequence that says stop, it gently lets go of the DNA and releases the finished RNA strand. The DNA closes calmly back into its double helix. The reading is done. The new RNA strand is called a messenger RNA, or mRNA for short. It is a soft, mobile copy of the gene that was just transcribed. It carries the message out of the nucleus into the patient workshop of the cell. Before the mRNA leaves the nucleus, it is gently edited. Small stretches called introns are cut out, and the remaining stretches called exons are softly spliced together. This is done by a small molecular machine called the spliceosome. A small protective cap is added gently to one end of the mRNA, and a long, soft tail of a nucleotides is added to the other end. The cap helps the mRNA be recognized. The tail helps it last longer before being broken down. Now, the edited mRNA is ready to leave. It slips quietly out of the nucleus through a small nuclear pore into the soft, warm cytoplasm, where the work of building proteins is done. In the cytoplasm, the mRNA will meet a ribosome, and the message it carries will be turned into a small, soft chain of amino acids, a protein. We will come to that very soon. Many genes are being transcribed in the nucleus right now. At any moment, in any one cell of you, several hundred RNA polymerases are quietly reading, each one writing a soft, temporary copy of a different gene. Which genes are being read depends on what the cell needs. In a brain cell, the genes for neurotransmitters are being read. In a stomach cell, the genes for digestive enzymes are being read. The same library, but the gentle selection differs. Tonight, while you rest, this quiet reading is happening very calmly in nearly every cell of you. mRNA copies are being made, edited, and gently sent out into the cytoplasm. The library is being used. The library is being kept alive. The messenger RNA, mRNA, leaves the nucleus carrying a soft copy of one gene's instructions. Out in the cytoplasm, it will find a small molecular factory called a ribosome, and the instructions will be slowly read into a protein. A ribosome is a small two-part structure made of RNA and protein, all wrapped softly together. There is a large subunit and a small subunit, and they come together gently around the mRNA when they are ready to start reading. Once the ribosome is assembled on the mRNA, it begins to read the message, but not letter by letter. Instead, it reads 3 letters at a time. Each soft group of 3 letters is called a codon. Each codon spells out one of 20 small molecules called amino acids. Amino acids are the building blocks of proteins. There are about 20 of them in living cells, each one with slightly different soft chemistry. So, as the ribosome reads each codon of the mRNA, it adds the corresponding amino acid to a growing chain. Codon by codon, amino acid by amino acid, the chain grows slowly out of the ribosome. To bring the right amino acid to each codon, the cell uses small soft helper molecules called transfer RNA, tRNA. Each tRNA molecule carries one specific amino acid on one end, and on the other end has a small soft loop that recognizes one specific codon. As the ribosome moves along the mRNA, the matching tRNA molecules drift in, gently dock at the current codon, deliver their amino acid to the growing chain, and then drift away again to be reloaded. The growing chain of amino acids is a young protein. As the ribosome adds each new amino acid, the chain gets a little longer and starts folding gently on itself in the soft, warm cellular fluid. The translation continues until the ribosome reaches a special soft codon called a stop codon. There are 3 of them, UAA, UAG, and UGA, and any of them tells the ribosome to stop and release the finished protein. Once the ribosome reaches a stop codon, the new protein is released into the cytoplasm. The ribosome itself drifts apart into its 2 subunits and is ready to be reused on another mRNA at another time. Translation is happening in vast numbers all over the cell. At any one moment, a single cell may have several 100,000 ribosomes, each one slowly reading an mRNA, each one building a soft new protein. Some ribosomes drift freely in the cytoplasm. Others are attached to a soft system of membranes called the endoplasmic reticulum. Proteins built on those membrane-bound ribosomes are sent gently into other parts of the cell or out of it entirely. The reading itself is fast for the small scale, but slow for us. A ribosome reads about 10 codons every second. A protein of 300 amino acids takes about 30 seconds to build, 30 soft seconds for one whole protein. Once the protein is released, it folds further, gently into its final three-dimensional shape. Sometimes other proteins called chaperones help with the folding. Once the shape is right, the new protein is ready to do its quiet work. And the proteins built this way are the workers of the cell. They build structures, carry molecules, catalyze reactions, send signals, defend against intruders. Almost everything the cell does is done very softly by proteins. Tonight, in every cell of you, thousands of new proteins are being built right now. Ribosomes are slowly reading. tRNAs are quietly delivering. Chains are gently folding. The patient work of being you is being made molecule by molecule while you rest. When a protein first comes off the ribosome, it is just a long, soft chain, a string of amino acids in a particular patient order. It is not yet useful to do its job. It has to fold gently into a very specific three-dimensional shape. The shape a protein folds into is determined entirely by the soft order of its amino acids. Some amino acids are slightly oily and tend to gather quietly in the middle of the fold. Others are slightly attracted to water and tend to face the outside. Some amino acids attract each other very gently through soft chemical bonds. Some repel each other. Some are large and bulky. Others are small and quiet. All of these soft local preferences add up very patiently into one particular three-dimensional shape. The folding usually starts very quickly, within milliseconds of the chain coming off the ribosome. The chain first forms small, soft local patterns called alpha helices and beta sheets. Then these patterns gently fold into larger arrangements. Alpha helices look like small, soft springs, sections of the chain coiling gently around an axis. Beta sheets look like soft, folded ribbons, sections of the chain running side by side, joined by gentle hydrogen bonds. These small, soft patterns combine into the overall fold of the protein. Some proteins look like soft, compact balls. Some look like long, curling ribbons. Some look like delicate tools with quiet, moving parts. Most small proteins can fold by themselves, very calmly in the soft cellular fluid. The amino acid sequence carries all the information needed. The shape is, in a quiet way, already implied in the order of letters in the gene. Larger or more complicated proteins often need a little help. There are special helper proteins called chaperones that gently guide the folding, keeping the new chain from tangling or sticking incorrectly to itself. Some chaperones look like soft barrel shapes. A misfolded protein can be drawn quietly into the barrel, given a gentle moment of seclusion in which to refold correctly and then released again into the fluid. Once a protein is fully folded, its shape determines what it can do. The shape is its function. A folded enzyme has a small, soft pocket exactly shaped to grip one specific other molecule. A folded antibody has a quiet tip shaped to recognize one specific intruder. The shape is everything. Sometimes proteins fold incorrectly. When this happens, the cell usually catches the mistake. Misfolded proteins are either softly refolded by chaperones or quietly broken down by small molecular machines called proteasomes, and their amino acids are gently recycled. Occasionally, a misfolded protein escapes all of this gentle quality control. In a few rare quiet diseases, misfolded proteins can clump together over time. But in most cells, most of the time, the folding is exquisitely accurate. Some proteins fold quickly into one shape and then stay there for their whole short life. Others have moving parts and can shift gently between different soft shapes depending on what they are doing, opening and closing like quiet, tiny doors. Many proteins also pair up with each other in small, soft groups. Hemoglobin in your blood, for instance, is made of 4 separate protein chains that fold gently together into one larger combined shape, with 4 little quiet pockets for carrying oxygen. Every second, in every cell of you, an enormous number of proteins are being folded. Most of them get it right. The ones that do not are quietly recycled. The whole patient system runs softly, day and night, without your needing to notice any of it. Tonight, while you rest, the folding continues. New chains are coming off ribosomes. Chaperones are gently helping. Proteasomes are quietly recycling. The slow, patient, three-dimensional sculpture of you is being made fold by fold, molecule by molecule while you sleep. DNA is durable but not invulnerable. Every single day, in every single cell of you, the DNA is damaged in small, soft ways. Sunlight breaks a bond. A chemical accidentally bumps into a base. A copy enzyme misreads a letter. Damage is constant. If none of this damage were ever fixed, your DNA would slowly degrade. But the cell has a small, soft crew of repair proteins that move quietly along the DNA, finding damage and gently mending it. There are several different kinds of damage and several different soft repair pathways. Each one is suited to a particular kind of problem, and each one is, in its own way, patient and careful. The most common kind of damage is a single misread letter, just one base, where it shouldn't be. The cell's mismatch repair system gently recognizes that one of the 2 strands no longer matches the other and fixes the soft mistake. Another common kind of damage is when a base becomes slightly chemically modified by sunlight, by a reactive molecule, or just by patient time. A small, soft enzyme called a glycosylase comes along, recognizes the damaged base, and snips it out. Then a different enzyme cuts the soft sugar phosphate backbone at the gap, and a polymerase puts in the correct new base. A ligase seals the gap. The DNA is mended almost as if the damage had never happened. Some kinds of damage are larger, for instance. When ultraviolet light from the sun fuses 2 adjacent bases into an unusual soft bump, special enzymes recognize the bump, cut out a small stretch of DNA around it, and patiently rebuild the stretch correctly. The most serious kind of damage is when both strands of the DNA are broken at once. A double-strand break. This is rare but very dangerous. because there is no intact template to read from when fixing it. The cell has two soft pathways to fix this. One pathway, called homologous recombination, very carefully uses the matching chromosome from the other parent as a template. It is slow and accurate. The other pathway, called non-homologous end joining, simply glues the two broken ends back together. It is faster but can lose small, soft pieces of the sequence. Among the most important repair proteins in your cells are two called BRCA1 and BRCA2. They are specialists in the careful repair of double-strand breaks. When they work well, the DNA is gently kept safe. When they cannot, breaks can accumulate. Another quiet but powerful repair protein is called p53. It is sometimes called the guardian of the genome. When DNA damage is too severe to repair, p53 gently tells the cell to pause its growth, or sometimes to softly self-destruct to prevent damaged DNA from being passed on. The total amount of repair happening in your body every second is hard to picture. In every cell, every day, thousands or tens of thousands of small repair events take place. Most go entirely unnoticed. None of them require your attention. When repair systems are working well, the DNA remains remarkably stable. The same long, quiet library that you were born with is mostly the same library you have today. Across decades, across nights of sleep, the library is being kept. When repair systems sometimes fail to keep up, through age or through specific damage to the repair genes themselves, the damage can slowly accumulate. Over long times, this is part of how cancers can softly begin, but for most cells, most days, the system holds. It is a small wonder that so much soft repair happens constantly with so little fanfare. The cell does not announce it. The repair just goes on quietly in nearly every cell, nearly every moment for your entire life. Tonight, while you rest, the repair crew is working in every cell of you. Damage is being recognized, sites are being snipped, polymerases are filling in, ligases are sealing. The library of you is being kept faithfully, page by patient page, while you sleep. No matter how good the repair, a few small changes do slip through. Not many, about 1 in every 10 billion letters per copying event. But across deep time, across many soft generations, these small slips gather gently into the patient differences between living things. A mutation is just a small change in the DNA sequence. Most of them happen for very ordinary reasons. A polymerase made a quiet mistake. A base was damaged and not perfectly repaired. A small piece slipped, or was added, or was lost. The simplest kind of mutation is a single letter change. One base where another base should be. We call this a point mutation or a substitution. It is the most common kind. Sometimes a single letter is added where there was none, an insertion. Sometimes a single letter is removed, a deletion. These are smaller in size but can sometimes shift the soft reading frame of the whole gene, changing all the codons downstream. Larger mutations can also happen more rarely. Whole stretches of DNA can be duplicated or inverted or moved to another part of the genome. The chromosomes themselves can occasionally exchange pieces with each other in a soft translocation. Most mutations have no effect at all. They land in stretches of DNA that do not code for proteins, or they change a codon to a different codon that happens to spell the same amino acid. We call these silent mutations. They are gentle, invisible. Some mutations are harmful. They change a critical amino acid in a protein, and the protein no longer folds or functions correctly. These mutations in important genes can cause disease, though most are quietly filtered out across generations. And occasionally, very rarely, a mutation is gently helpful. It changes a protein in a way that lets the living thing it is in survive a little better in some quiet new circumstance. These are the mutations that, across the long ages, slowly shape evolution. In the very early embryo, mutations that occur are quietly passed on to every cell of the developing body. These are the mutations that contribute to who you become. Most of these small changes are gentle individual differences. Mutations in your reproductive cells, eggs or sperm, can be passed on to your children. These are the mutations that very slowly accumulate across generations, becoming the patient differences between families, populations, species. Mutations in your other cells, skin, blood, gut, brain, stay with you but are not passed on. They are part of the slow individual mosaic that develops in everybody over a lifetime. Most are quiet and harmless. The pace of mutation is very slow at the individual level. In each generation, a person inherits about 70 new mutations on average from their parents, 70 small soft differences in 3,000 million letters. 70 new mutations per generation, most invisible, but across 100 generations, 3,000 or 4,000 years, that gentle drift adds up to perhaps 7,000 small soft differences. Across 1,000,000 generations, the differences add up to whole new species. Every variation between two living things, every brown eye, every blue eye, every freckle, every gentle tendency and temperament, is somewhere written in this patient's slow accumulation of small, soft mutations across deep time. And most of these mutations are not anyone's doing. They happen by quiet chance, by the soft chemistry of bases, by the patient stochastic limits of even the best repair systems. They are part of what it means to be alive at all. Tonight, somewhere in your body, a small new mutation is happening. It is almost certainly silent. It will not matter to you. It is part of the slow patient texture of being a living thing while you rest in the dark. At each end of each chromosome, there is a soft repeating sequence called a telomere. It is a kind of gentle protective cap made of the same 6 letters repeated many thousands of times, TTAGGG, over and over. Telomeres exist because of a small, soft, technical problem with DNA replication. When DNA polymerase copies a strand, it cannot quite finish copying right at the very end. A few letters at the tip always get left out. If the very ends of the chromosomes carried important information, this small soft loss would be a problem, so instead the ends carry the patient repeating telomere sequence. The lost letters at each copying are just telomere letters, and the important genes inside are protected. Each time a cell divides, the telomeres at the ends of its chromosomes get a little shorter. A few dozen letters every division. Slowly, very slowly, the soft caps gently shrink across the lifetime of a cell. When the telomeres get very short, too short to keep the chromosome ends gently protected, the cell senses this and quietly stops dividing. The cell enters a calm state called senescence and rests there for the rest of its life. This patient shortening is, in part, why many of our cells have a limited number of divisions they can make. It is one of the soft built-in clocks of being multicellular. It is also part of why we slowly age. There is a small enzyme called telomerase that can gently rebuild the telomere ends, adding the soft T-tag repeats back on. In most of your adult cells, telomerase is gently switched off, but in a few special cells like stem cells and reproductive cells, it stays softly active. In your stem cells, telomerase keeps the telomeres long, so those cells can keep dividing throughout your life, gently regenerating the tissues that need constant renewal: your skin, your gut lining, your blood. In your reproductive cells, eggs and sperm, telomerase is also gently active, so the chromosomes that will be passed to your children have their telomeres restored to full length at the start of their next life. Telomere length varies between people, between cell types, between ages. Babies are born with longer telomeres on average. Older people have shorter ones. Some lifestyle factors gently affect telomere length: sleep, exercise, stress. The telomeres also serve another quiet function. They prevent the cell from mistakenly treating the natural end of a chromosome as if it were a dangerous DNA double-strand break. Without the soft protective cap, the cell would try to repair the end, which could be very damaging. To do this protective job, the telomere DNA is gently wrapped in a small set of special proteins called the shelterin complex. These soft proteins keep the chromosome end hidden and quiet, signaling to the rest of the cell that everything is fine. Telomeres are present in every chromosome of every nucleated cell of you. 46 chromosomes per cell, each with 2 soft telomere caps, 92 patient little shelters per cell, gently keeping each chromosome end safe. The discovery of telomeres and telomerase took most of the 20th century to unfold. Many quiet scientists across many soft decades pieced together how the ends of chromosomes are kept safe. It was in itself a patient piece of work. There is something gentle and reassuring in this. Even the very ends of your chromosomes are softly cared for. Even the soft buffering region is in turn buffered. The genome takes very, very good care of itself. Tonight, in every cell of you, the patient ends of the chromosomes are gently capped. The shelterin proteins are softly in place. Most of the telomeres are not changing at all right now. The library has its quiet bookends, and the bookends are holding. Almost all of your DNA lives quietly in the nucleus of your cells, but not quite all. There is a small second library, a tiny separate piece of DNA that lives somewhere else entirely. It lives inside the mitochondria. Mitochondria are small bean-shaped structures inside your cells. They are the cell's quiet power plants. They take in oxygen and small food molecules and gently turn them into a usable energy currency called ATP, which the cell uses for almost everything. Each of your cells has between a few dozen and several thousand mitochondria, depending on how much energy the cell needs. Heart muscle cells, which work constantly, have many. Skin cells have fewer. Each mitochondrion is its own quiet, small organelle. Inside each mitochondrion, there is a small circular molecule of DNA. Not the long, straight chromosomes of the nucleus, but a soft little loop, only about 16,000 letters long. This is your mitochondrial DNA. 16,000 letters is tiny compared to the 3,000 million letters of your nuclear DNA. The mitochondrial DNA carries only 37 genes, instructions for 13 proteins, 22 tRNAs, and 2 ribosomal RNAs, all involved in the patient work of energy production. The reason mitochondria have their own DNA goes back almost 2,000 million years. Long ago, a small ancient bacterium gently joined with a larger ancient cell, and instead of being digested, it stayed inside. Over time, the bacterium became a permanent quiet partner, the mitochondrion. Over almost 2,000 million years, most of the original bacterial DNA was either lost or quietly transferred to the nucleus of the host cell, but a small, soft loop of essential genes stayed inside the mitochondrion, where it still lives today. Mitochondrial DNA is inherited very differently from your nuclear DNA. The nuclear DNA you have came half from your mother and half from your father, but your mitochondrial DNA came entirely from your mother. This is because when an egg is fertilized by a sperm, the egg contributes all the cytoplasm and therefore all the mitochondria, while the sperm contributes only its nuclear DNA. So the mitochondrial DNA inside you came from your mother and from her mother and so on, in an unbroken line of mothers. Because mitochondrial DNA is passed only from mother to child, it serves as a quiet marker of maternal ancestry. By comparing the small differences in mitochondrial DNA between people, scientists can softly trace back maternal lineages across many generations. Following these soft mitochondrial signals, scientists have traced the maternal ancestry of all living humans back to a single woman who lived in Africa around 200,000 years ago. She is sometimes called Mitochondrial Eve. She was not the only woman alive then. She is just the soft tip of the only maternal line that has continued unbroken into every human alive today. Every person alive tonight carries mitochondrial DNA that is, very softly descended from that one quiet ancestral woman. The same circular loop, slightly modified across 200,000 years of patient generations. Mitochondrial DNA also changes slightly faster than nuclear DNA because mitochondria don't have quite as many of the soft repair systems that the nucleus has. So mitochondrial differences between populations are useful for tracing more recent ancestry. Some rare diseases are caused by mutations in mitochondrial DNA. Because every cell has many mitochondria, the effects can be soft and varied, different cells inheriting slightly different mixes of normal and mutated mitochondria. Each mitochondrion can divide on its own, gently inside the cell. They split softly in half, copy their small DNA and become two, so the population of mitochondria in your cells is always slowly being renewed even when the cell itself is not dividing. Tonight, inside every cell of you, hundreds or thousands of small mitochondria are quietly doing their slow work. Each one holds a soft circular loop of DNA that is very gently your mother's and her mother's all the way back. The other library is also being kept. Every cell in your body has the same DNA, yet a brain cell looks nothing like a liver cell. A skin cell does nothing like the work of a heart cell, the same library, but read in very different patient ways. How does the cell know which stretches to read and which to keep quiet? The answer is a soft second layer of information that lives on top of the DNA itself. We call this the epigenome, meaning above the genome. The DNA letters are the same in every cell, but the soft chemical marks on top of them differ. These marks are not changes in the DNA letters. They are small, soft chemical tags that quietly say things like "read this stretch loudly," or "keep this stretch silent," or "remember that you opened this region yesterday." There are two main kinds of epigenetic marks. The first kind lives directly on the DNA. A small, soft chemical group, a methyl group, made of one carbon and three hydrogens, can be gently attached to certain C bases in the DNA sequence. DNA methylation, as this is called, usually quietly silences the stretch of DNA where it sits. The methyl marks tell the cell, "This region is closed. Do not read it. Keep it gently tucked away." The second kind of mark lives on the histone proteins that spool the DNA. Histones have soft tails that stick out from their round shape, and these tails can carry many different small chemical marks: acetyl, methyl, phosphate, and others. Different patterns of marks on the histone tails mean different things. Some marks open the chromatin and invite reading. Others close the chromatin and silence the region. The combination of marks at a given place is sometimes called the soft histone code. These epigenetic marks are gently maintained across cell divisions. When a cell divides, the daughter cells inherit not just the DNA, but also most of the soft marks on it. So a skin cell daughter usually stays a skin cell. A brain cell daughter usually stays a brain cell. During early development, when a single fertilized egg becomes a whole embryo, the epigenetic marks are quietly set and reset many times. Each different tissue gets its own soft pattern of marks. This is how cells with identical DNA become so wonderfully different. Epigenetic marks can also change in response to a person's experiences. Diet, stress, exercise, sleep, exposure to smoke can all gently shift the soft marks on certain genes. Most of these shifts are small and reversible. Some are longer lasting. Some epigenetic marks may even pass from parent to child. This is rarer and more controversial in humans, but in some small studies, soft echoes of a parent's experiences seem to gently appear in the next generation's gene expression. It is still a quiet area of active research. The total number of epigenetic marks across the human genome is enormous. Millions of methylation sites, dozens of different possible histone modifications. The full soft epigenome is even more complex than the genome itself. In one sense, the genome is the long, quiet library, and the epigenome is the soft list of which books are currently checked out, which are open, which are dusty on the shelf. Both layers are needed to know what the cell is actually doing. Epigenetic patterns can drift slowly across a lifetime. Old tissues sometimes have somewhat altered epigenetic marks compared to young tissues. This soft drift is part of what we call epigenetic aging, and it is a quiet topic of much current research. The fact that the same DNA can do such different things in different cells is, in a quiet way, one of the great wonders of biology. The same library, read in countless different gentle ways, producing the soft variety of a whole living being. Tonight, in every cell of you, the soft epigenetic marks are in place. The skin cells are reading their skin genes. The neurons are reading their neuron genes. The library is being selectively, gently, patiently read, and the soft layer of marks above it is being quietly kept. Inside almost every cell of you, there is a small, soft molecular clock. It runs on a roughly 24-hour rhythm. It tells the cell what time of day it is, even when there is no light. It is built from a small set of patient genes called the circadian clock genes. The main clock genes have soft names, period, called per, cryptochrome, called cry, and the soft pair clock and BMAL1. Together they form a small patient feedback loop that ticks softly through the day. Early in the morning, the clock and BMAL1 proteins gently bind together and turn on the per and cry genes. The cell begins to make per and cry proteins. Over the day, these proteins gradually accumulate in the cytoplasm. By late afternoon and evening, enough per and cry proteins have built up that they begin to drift back into the nucleus and gently bind to clock and BMAL1, telling them to stop turning on the per and cry genes. The system is gently switched off. Through the night, the per and cry proteins slowly degrade. By dawn, there is very little left of them, and clock and BMAL1 can begin the cycle again. One quiet round of the loop takes about 24 hours. This patient loop runs in nearly every cell of your body. Skin cells, liver cells, heart cells, almost all of them have their own internal small clock running through this same soft cycle. All of these distributed cellular clocks are gently coordinated by a single small master clock in the brain, in a quiet region called the suprachiasmatic nucleus, or SCN. The SCN is a soft cluster of about 20,000 neurons, deep in a structure called the hypothalamus. The SCN receives soft light signals from your eyes through a special pathway. When morning light reaches your retina, small soft signals travel into the SCN and gently reset the master clock to match the outside day. The SCN then sends soft signals out to the rest of the body, through nerves, through hormones, through subtle changes in body temperature, telling all the other cellular clocks what time the master clock thinks it is. One of the soft messengers is the hormone melatonin. As evening begins, the pineal gland in your brain starts to release melatonin into your blood. It quietly tells the rest of your body that night is coming and that it is gently time to slow down. Your sleep itself is governed by these soft circadian rhythms. Body temperature drops slowly in the evening, heart rate slows, hormone patterns gently shift, the body very patiently prepares itself for sleep. None of it requires your effort. Different people have slightly different versions of the circadian genes. These soft differences explain part of why some people are gentle morning larks and others are quiet night owls. It is in some small part genetic. When circadian rhythms are gently disrupted by jet lag, by shift work, by long screen exposure in the late evening, the soft clock genes can become misaligned. Many small downstream effects follow. The body's quiet rhythm needs its regular soft cues. Sleep itself, when it happens, is when many quiet maintenance and repair processes are most active. The brain's soft cleaning system runs more strongly. DNA repair in many tissues runs faster. The body uses the patient quiet of night for soft inner work. So when you sleep tonight, the circadian clock genes are doing exactly what they have always done. Clock and BMAL1 are quiet now. Per and cry are gently breaking down, By morning, the cycle will softly begin again. Your sleep tonight is not an absence. It is a soft, active phase of a long, quiet rhythm that has been running in every cell of you, every single day of your life. The clock is ticking very gently. You only have to rest. DNA is the way one generation gently hands itself to the next, from parent to child, from grandparent to grandchild, across all the soft generations that have ever lived. This is heredity. When a person has a child, they each contribute half of the child's nuclear DNA. The mother contributes 23 chromosomes in her egg cell. The father contributes 23 chromosomes in his sperm cell. The two soft sets come together to make the full 46. The egg and the sperm are made by a special soft kind of cell division called meiosis. Meiosis is different from normal cell division. Instead of producing two identical daughter cells, it produces 4 soft daughter cells, each with only half the usual number of chromosomes. During meiosis, the matched pairs of chromosomes from your two parents gently pair up. While they are paired, they softly exchange small stretches of DNA with each other. This soft exchange is called recombination, or crossing over. Recombination is the reason siblings are not identical. Each egg cell or sperm cell carries a slightly different gentle mix of grandparental DNA. Each new generation is a soft shuffle of the genetic deck. Beyond recombination, the chromosomes also sort themselves softly into the egg and sperm in different combinations. The 23 from each parent can shuffle in millions of different gentle orders. Every egg and every sperm is uniquely arranged. When the egg and sperm meet at fertilization, they form a single soft cell with a unique combination of DNA that has never existed before. This single quiet cell will, across the slow patient months of development, become a whole person. Some traits, of course, are visible. The shape of an eyebrow, the color of an eye, the soft way a smile spreads. Many of these visible traits are influenced by many genes together, with soft contributions from each. Other traits are determined by a single gene, often with a clean inheritance pattern. Eye color was once thought to be like this, though it turns out to be more complicated. A few soft traits, like certain blood types or some genetic conditions, really do follow simple Mendelian patterns. Many traits are also influenced by the environment as well as by the genes. Height, for instance, has a strong genetic component, but is also shaped by nutrition and other gentle factors during childhood. Most traits are a soft combination of inheritance and experience. The soft pattern of inheritance for any single trait depends on whether the gene is dominant or recessive. A dominant version of a gene shows its effect even when you have only one copy. A recessive version only shows its effect when you have 2 copies, one from each parent. Some genetic conditions appear in this soft pattern. If both parents carry one copy of a recessive variant, they may show no signs themselves, but their child may inherit 2 copies and develop the condition. Many gene variants work this way. The chromosomes themselves can sometimes be inherited in slightly different numbers, most commonly with 23 pairs, but occasionally with an extra chromosome, like in Down syndrome, where there's a 3rd copy of chromosome 21. These variations are part of human diversity. DNA testing today can read your full sequence, all 3,000 million letters, for a relatively small cost. From this, soft echoes of ancestry, of medical risk, of family connection can be quietly read. The library inside you can now, in some part, be opened. And yet, for almost all of human history, none of this was known. People simply lived, gently had children, watched their faces and ways resemble their parents. The DNA was doing its quiet, patient work all along, in complete silence. Tonight, the DNA inside you is half from one parent, half from another, with small, soft pieces from grandparents and great-grandparents, all gently mixed by generations of slow, patient recombination. You are a soft current within the longest river there is. Every living thing on Earth uses DNA. Every tree, every bird, every quiet fish in the deep sea, every soft mushroom in the forest, every patient bacterium in the soil, all of them, the same molecule, the same 4 soft letters. Not just the same molecule, the same exact way of using it. Every living thing on Earth uses the same code. The same 3-letter codons spell the same amino acids in a bacterium, in a mushroom, in a redwood tree, in you. This shared code is, very softly, a deep clue. It tells us that every living thing on Earth tonight is descended from the same patient distant ancestor. A single soft starting cell, almost 4,000 million years ago, from which all of life has slowly grown. That distant ancestor is sometimes called Luca, the last universal common ancestor. We do not know exactly what Luca looked like, but by comparing the soft DNA of modern creatures, we can quietly reconstruct some of what Luca must have been like. Luca used DNA. It used the same 4 letters. It used the same triplet codon code. It had ribosomes. It had at least some of the same patient enzymes that all life still has today. The basic equipment of being alive was already in place. From Luca, life slowly diversified. Some descendants became bacteria, others became archaea. Much later, some cells merged with others in soft endosymbiosis and became more complex cells, eukaryotes, with nuclei and mitochondria. From early eukaryotes, life branched again, slowly. Some became plants, some became fungi, some became animals. Each soft branch carried forward the same patient DNA molecule, the same 4 letters, the same universal code. If you compare your DNA letter by letter with the DNA of a chimpanzee, about 98% of the letters match. Your common ancestor with a chimpanzee lived about 6 million years ago. That much soft sequence has been preserved across that distance. Compare your DNA with a mouse. About 85% of your letters match. The common ancestor of humans and mice lived about 80 million years ago, around the soft end of the dinosaur age. That much resemblance is still here. Compare your DNA with the small, soft fruit fly. About 60% of your genes have a clear, soft equivalent in the fly. Many of the basic genes for being a complex animal, for building bodies, growing eyes, forming nerves are gently shared. Compare your DNA with a banana plant. About 50% of your genes have a soft equivalent in the banana. The basic machinery of being alive, for energy, for division, for patient maintenance, is shared between you and a banana. Compare your DNA with the DNA of bacteria. The letters are still mostly recognizable. The basic enzymes for copying DNA, for reading DNA, for building proteins, all look obviously like the equivalents in bacteria. Even at this soft distance, we are still cousin. It is in a quiet way very beautiful. Every creature alive tonight, every owl in the dark, every patient tree growing slowly in the soft cold, every soft fish in the depths of the sea is reading the same kind of book, letter by letter, codon by codon. The DNA inside you tonight is a soft current of a river that has been flowing for almost 4,000 million years, from Luca, through bacteria, through ancient soft single-celled ancestors, through the first multicellular things, through fish, through small ancient mammals, through long lines of patient ancestors, all the way to you. And it will flow on past you. Whatever happens in your soft life, the DNA molecule itself, the 4 letters, the universal code, will continue gently into the future in the cells of your descendants and in the cells of all the other patient creatures still to come. Tonight, in every cell of you, the same 4 soft letters that have spelled out all of life since the beginning are doing exactly what they have always done: copying, reading, folding, resting, the one quiet library still open while you sleep. Right now, while you rest, the DNA inside you is doing many quiet things. It is being read, it is being copied, it is being repaired, it is being gently maintained. None of it requires your attention, and none of it ever has. In the cells of your skin, the very cells just under the surface, on the back of your hands, on your forehead, DNA is being copied right now. Skin cells divide patiently to replace the soft outer layer. The library is being duplicated over and over. In the lining of your gut, DNA is being copied even faster. Gut cells live only a few days before being gently replaced. While you sleep, the gentle factory in your intestines is renewing itself, copying its quiet library again and again. In your bone marrow, deep in the soft hollows of your bones, stem cells are dividing patiently to produce new blood cells for tomorrow. Each division is a quiet, careful copying of the long library. The blood you will use tomorrow is being made tonight. In your brain, even now, neurons are reading genes for the soft molecules of sleep, for the proteins of memory consolidation, for the gentle maintenance of synapses. The library is being selectively read for the patient work of the night. In your liver, in your kidneys, in your spleen, almost every organ has cells reading their own particular patient subset of the library, doing the slow chemistry of being alive, all of it gentle, all of it without your attention. In every nucleus of you, the same 6 picograms of soft DNA sit, gently folded, patiently held in place by histones, softly capped at the ends by telomeres, marked here and there by quiet epigenetic tags. The library complete in every cell. In every mitochondrion of you, a small, soft, circular DNA loop is also gently being read and copied. The other library, the maternal one, is also patiently at work in the dark. Tonight, it too is being kept. If you could very quietly listen to your DNA tonight, what you would hear would be the softest possible thing. Bonds gently breaking and reforming, polymerases gliding, tiny molecular machines doing their patient work in the warm, dark cytoplasm, a long, quiet hum of life. The exact arrangement of letters inside you is unique. No one else in the world has exactly this combination, except possibly an identical twin who would still have small, soft differences from accumulated mutations. You are, in the long, quiet way, alone in your particular sequence. And yet most of those letters are deeply shared. Nearly everyone alive shares almost all of your sequence, and almost every other living thing shares many of the core patient regions. You are uniquely arranged, yet softly made of the same shared material as all of life. The DNA inside you is also very old. Some of the soft stretches in your genes have been preserved nearly unchanged for hundreds of millions of years. Some of the core sequences for being alive are essentially the same in you as they were in your distant ancestors before there were vertebrates at all. And while you sleep, a small number of new soft mutations are arising somewhere in your body. Most will be invisible, most will be silent. They are simply part of the gentle, ongoing texture of being alive at the molecular level. Your DNA does not know that you are tired, that you have had a long day, that you are about to sleep. It does not need to know. It simply does the same patient work it has always done, regardless of the larger world around it. There is something quietly comforting in this. Whatever the day brought, whatever tomorrow may bring, the molecular you, the very long, quiet library, is calmly doing what it has always done. The library is being kept. The library is always being kept. Tonight, while you breathe softly, your library is gently open. It is being read in some places, copied in others, rested in most. It is very quietly, simply being itself, and being itself is being you. It is, in a soft way, hard to believe, inside every single cell of you, 2 meters of a patient twisted molecule holding the quiet instructions for being you, copied faithfully across nearly 4,000 million years. 4,000 million years is a very long time. Long enough that the continents have moved many times, long enough that the oceans have risen and fallen many times, long enough that whole quiet kinds of life have appeared and gently disappeared. Through all of it, the DNA molecule has stayed, its basic shape, 2 strands wound gently around each other, its basic alphabet, 4 small letters, its basic way of copying, soft pairing across 2 patient strands. None of this has fundamentally changed. Every living thing alive tonight is the soft tip of an unbroken chain of molecular copying that reaches all the way back to Luca. Every cell of you is part of a quiet lineage that has in some way never paused. Your great-grandmother's cells copied their DNA gently into her eggs. One of those eggs became your grandmother. Your grandmother's cells copied their DNA gently into her eggs. One of those eggs became your mother. Your mother's cells copied their DNA gently into her egg. One of those eggs became you. This chain of patient copying goes back mother to mother for thousands of generations, for tens of thousands of generations, for hundreds of thousands. The copying has never broken. The library has never been entirely lost. The thread has always continued. And the chain goes far further back. Before there were humans, before there were primates, before there were mammals, before there were vertebrates, before there was anything we would now recognize as an animal at all. The copying just kept happening, generation after gentle generation, for nearly 4,000 million years. The 4 small letters did not change. The shape of the double helix did not change. The patient pairing rules did not change. The basic enzymes for copying and reading and repairing have stayed remarkably the same across all of it. Of course, the particular arrangement of letters has changed. Tonight you carry a slightly different soft sequence from any creature that came before you. Every generation has added its own small differences. The patient molecule has been the same, but the patient text has slowly evolved. And tonight, somewhere in your cells, a new small mutation is happening. A tiny, gentle change in the long text. It is almost certainly silent, but it is part of the same patient process that has, across 4,000 million years, produced the soft variety of all life. There is a kind of soft wonder in this. The library inside you is older than the continents in their current positions. It is older than the dinosaurs. It is older than oxygen in the atmosphere. It is, in molecular terms, almost as old as the Earth itself. And the library is shared. Whatever differences exist between you and the patient owl asleep in a tree across the world, between you and the soft fish at the bottom of the sea, between you and the quiet bacterium in the soil, all those differences are small variations on the same gentle theme. If you ever feel alone, the molecular truth is that you are connected to every living thing on Earth by the same shared patient four-letter library that all of life uses. Every breath of every other creature is, in some quiet molecular way, related to your own. None of this is something you have to do. None of it is something you have to remember tomorrow. The patient long library inside you has been being itself very quietly for your whole life. It has been keeping you. It will keep keeping you. So while you rest tonight, the DNA inside you is just doing what it has always done. The soft, slow miracle of molecular self-copying going on in the dark in nearly every cell of you without any effort or attention. There is nothing you need to do. The library is being kept. The library is always being kept. You are part of a long, soft, patient wonder, and the wonder is doing very well without your help. And so we have come very softly to the end of our quiet journey, from the first idea of inheritance through the shape of the double helix, the four small letters, the patient copying, the gentle reading, the long ancestry, all the way back very softly to your sleeping body in the dark. Nothing about any of this needs to be remembered. The molecule is doing all of it for you without your attention. It always has, it always will, for as long as you are alive. Inside every cell of you tonight, the soft library is open. Some pages are being read, most are gently resting. The patient chromosomes are folded, the telomeres are capped. The repair crew is on its quiet shift. Everything is being kept. Thank you for staying with us this long. If sleep is closer now than it was when we began, then this night did its work. Subscribing helps us return next week with another slow journey through the slow molecular library inside every living cell, recorded with the same patient voice you have been listening to tonight. Across the world tonight, in the cells of every living thing, the same four soft letters are quietly doing the same patient work. Trees, owls, whales in the deep sea, tiny bacteria in soil, and you breathing softly with the same shared molecular continuity in every cell. There is no part of this you have to manage. There is no page you have to turn. There is no copying you have to oversee. The molecule very patiently is doing it all in the dark, and it has been doing it for nearly 4,000 million years. Good night. Let yourself rest now. Somewhere tonight, inside a quiet cell in your fingertip, a soft molecule is being copied exactly as it was copied in your great-grandmother. Somewhere, a patient ribosome is reading 3 letters at a time. Everywhere on Earth, the same 4 quiet letters are spelling out the long, calm story of life while you rest. The world will keep going, with or without your watching. You can let go. Good night from the Circadian Institute.